Hello from Miss Brown. Today we have a screencast on DNA structure and code. Keep in mind, again, you can always stop and start this as we go. This is going to be a good bit of information, so I'm sure you'll have to stop it as we go. So DNA is the code of life, and it stands for this really large word, deoxyribonucleic acid. And number 2, all cells contain DNA. Now, eukaryotic cells, of course, have their DNA in their nucleus. It's what makes up chromosomes. And the shape of DNA is often described as a double helix, which we'll look at in just a second. So here we have it. It's like a ladder, then the ladder itself is twisted up. And you'll be doing a diagram of this in the next day or two. Now, what is it made of? So it's made of these things called nucleotide subunits. So the nucleotides have 3 parts. So look at number 5. The first part is a deoxyribose sugar. The second part is a phosphate. And the last piece is the nitrogen base. And there's 4 bases. It could be either adenine, guanine, cytosine, or thymine. And we'll often refer to these with their letters from here on out. The sides of this ladder are the sugar and phosphate, alternating every other one. And then the steps across the center where you would step, or the rungs, are the nitrogen base compounds. As you can see here, we see here's the adenine, thymine, guanine, cytosine. Here's where they go across. And up and down here we have the sugar and phosphate up the side arms of the ladder. Now, number 8, DNA purines are large nitrogen compounds. Those are adenine and guanine. And the DNA pyrimidines, they're the smaller pieces. They're thymine and cytosine. So now, if you'll notice, look how this works. A big piece, the A, goes with a smaller piece, the T. A smaller piece, C, will go with a bigger piece, G, and that helps them fit together. A and G could not fit together. They're both too large. And C and T wouldn't be large enough. So you have to have a big piece and a little piece. And specifically, this thing called the complementary base pairing rule, or Chargaff's rule. A always pairs with T, and C will always pair with G. And there'll be a video later explaining this a little more in depth. Now, what holds them together is a weak hydrogen bond that holds those together in the center, and then it's twisted, and that strengthens it. Now, how was this discovered? Well, it was first discovered by Mischer in 1869. So we've known about DNA for a while. We just didn't really know what the structure was. He actually obtained it. He got phosphorus, which was a white powder. It was white powder, and he didn't get phosphorus. It contained phosphorus, I should say. And it had a slightly acidic pH, which makes sense. Deoxyribonucleic acid. Okay. Now, our current model, I don't have a slide for this one, but our current model of DNA was determined by Watson, W-A-T-S-O-N, and Crick, C-R-I-C-K, in the 1950s. That's number 14. So now, let's talk about DNA and what it does when it gets ready to make more copies of itself. So under DNA replication, number 1, when DNA replicates, it makes a replica, which is a copy of itself. And why does it have to do this? So the cells can divide, like we learned about in mitosis. There has to be enough DNA, enough chromosomes to put in each new daughter cell. So it has to do that to divide. And there's 3 steps. First, that ladder that's twisted up must uncoil, and then it must unzip, and then it must copy itself exactly, because if it's not exactly the same, the cell will be different. And all this action is enzyme controlled. So the enzyme causes all this to uncoil, unzip. This pops open. Remember, these are weak bonds. It pops open. And then new pieces join in. And we'll be watching a video on this later so you can see kind of how it works a little better. Then new pieces. So you have one old piece as a pattern, and that brings in the new pieces that join up. And over here we have an old piece and a new piece. Okay? So this is what we call semi-conservative, which we'll get to in a second, but look at number 4. This enzyme that controls this, DNA polymerase. And of course, we've already learned that ACE means enzyme. So once this happens, it has to divide. We got to move on. There's too much genetic info being carried around. And this method, number 5, is what we call a semi-conservative method. It means we conserve half of it. So that means that DNA is half old and half new. So this is designed to reduce errors because you always have that good pattern to go by. Okay, so let's talk about this code. What's this all about? So understanding the code. All right, number 1, the DNA code is interpreted along one side of the ladder, 3 letters at a time. And these 3-letter combos are called triplet codons. And the triplet codons contain information to make proteins. All right, moving on to number 4. This is how DNA controls heredity. This message, DNA has this message to make proteins, to tell your body everything from your eye color to your height potential to your IQ maybe, even to how your body digests food, how your body handles sugar and insulin. So that's what's contained in this code. It's everything, everything. Sorry, I went backwards. So it controls heredity. And this heredity, look here at the top, at the last part of number 4, is called gene expression. Gene expression. So it's how you express. Like I express blue eyes. I express tall height. I express curly hair. Okay, so all things are how it's expressed. My genes that make me a unique individual and you a unique individual. So again, DNA is found in the nucleus of the eukaryotic cell. But these proteins are made out in the cytoplasm at something called ribosomes. Remember, ribosomes are the protein factories. And this is where they're made, on the ribosomes. And number 6, DNA makes a compound called RNA to help with protein synthesis. So how does it make this stuff called RNA? Well, number 1, under DNA, how DNA makes RNA. DNA makes RNA by a process called transcription. We'll talk about this first. Transcription. Now, this transcription, which means to make a copy, because scribe means to copy or write. So we're going to make a copy. So this is controlled by an enzyme, number 2, RNA polymerase. And it takes place in the nucleus where the DNA is. And this is the first of 2 steps. And we'll work on the first step first, of course. So RNA stands for ribonucleic acid. So let's look at number 5 and compare RNA and DNA. DNA is double-sided. RNA is single-sided. DNA contains deoxyribose sugar. RNA contains ribose sugar. DNA has the bases A, T, C, and G, but RNA has A, U, C, and G, and the U stands for uracil. So the U is going to substitute for T in the code system. Now, there's 3 kinds of RNA. There's messenger RNA. It's a little m and then the 3 big letters. That carries the message. It's the messenger. rRNA is what makes up ribosomes. It's ribosomal RNA. And tRNA is the one that I say goes out and does the shopping. It's the one that gets everything lined up and lines up these amino acids in translation, which we will be getting to in a few days. Okay, thank you.